Hello there, how are you? Welcome to another biology class. Today in part 2 we will talk about the genetic blueprint of organisms called DNA. Before we start, make sure you have subscribed to this channel and please be kind to like and share this video. Alright then, let us begin.
Nucleobase classification. The nucleobases are classified into two types, the purines, A and G, which are fused 5 and 6-membered heterocyclic compounds, and the pyrimidines, the 6-membered rings C and T. A fifth pyrimidine nucleobase, uracil U, usually takes the place of thymine in RNA and differs from thymine by lacking a methyl group on its ring. In addition to RNA and DNA, many artificial nucleic acid analogues have been created to study the properties of nucleic acids, or for use in biotechnology. Non-canonical bases. Modified bases occur in DNA. The first of these recognized was 5-methylcytosine, which was found in the genome of Mycobacterium tuberculosis in 1925. The reason for the presence of these non-canonical bases in bacterial viruses, bacteriophages, is to avoid the restriction enzymes present in bacteria. This enzyme system acts at least in part as a molecular immune system protecting bacteria from infection by viruses. Modifications of the bases cytosine and adenine, the more common and modified DNA bases, play vital roles in the epigenetic control of gene expression in plants and animals. A number of non-canonical bases are known to occur in DNA. Most of these are modifications of the canonical bases plus uracil.
Grooves, DNA major and minor grooves. Twin helical strands form the DNA backbone. Another double helix may be found tracing the spaces, or grooves, between the strands. These voids are adjacent to the base pairs and may provide a binding site. As the strands are not symmetrically located with respect to each other, the grooves are unequally sized. The major groove is 22 angstroms, 2.2 nanometers wide, while the minor groove is 12 angstrom, 1.2 nanometer in width. Due to the larger width of the major groove, the edges of the bases are more accessible in the major groove than in the minor groove. As a result, proteins such as transcription factors that can bind to specific sequences in double-stranded DNA usually make contact with the sides of the bases exposed in the major groove. This situation varies in unusual conformations of DNA within the cell, but the major and minor grooves are always named to reflect the differences in width that would be seen if the DNA was twisted back into the ordinary B form.
Base pairing. In a DNA double helix, each type of nucleobase on one strand bonds with just one type of nucleobase on the other strand. This is called complementary base pairing. Purines form hydrogen bonds to pyrimidines, with adenine bonding only to thymine and 2 hydrogen bonds, and cytosine bonding only to guanine and 3 hydrogen bonds. This arrangement of 2 nucleotides binding together across the double helix from 6-carbon ring to 6-carbon ring is called a Watson-Crick base pair. DNA with high GC content is more stable than DNA with low GC content. A Ho-Augustine base pair, hydrogen bonding the 6-carbon ring to the 5-carbon ring, is a rare variation of base pairing. As hydrogen bonds are not covalent, they can be broken and rejoined relatively easily. The 2 strands of DNA in a double helix can thus be pulled apart like a zipper, either by a mechanical force or high temperature. As a result of this base pair complementarity, all the information in the double-stranded sequence of a DNA helix is duplicated on each strand, which is vital in DNA replication. This reversible and specific interaction between complementary base pairs is critical for all the functions of DNA in organisms.
ssDNA versus dsDNAs. As noted above, most DNA molecules are actually 2 polymer strands, bound together in a helical fashion by non-covalent bonds. This double-stranded dsDNA structure is maintained largely by the intrastrand base stacking interactions, which are strongest for G-C stacks. The 2 strands can come apart, a process known as melting, to form 2 single-stranded DNA ssDNA molecules. Melting occurs at high temperatures, low salt and high pH. Low pH also melts DNA, but since DNA is unstable due to acid depurination, low pH is rarely used. The stability of the dsDNA form depends not only on the GC content, %G-C base pairs, but also on sequence, since stacking is sequence-specific, and also length. Longer molecules are more stable. The stability can be measured in various ways. A common way is the melting temperature, also called TM value, which is the temperature at which 50% of the double-strand molecules are converted to single-strand molecules. Melting temperature is dependent on ionic strength and the concentration of DNA. As a result, it is both the percentage of GC base pairs and the overall length of a DNA double helix that determines the strength of the association between the two strands of DNA. Long DNA helices with a high GC content have more strongly interacting strands, while short helices with high AT content have more weakly interacting strands.
In biology, parts of the DNA double helix that need to separate easily, such as the two-tau box and some promoters, tend to have a high AT content, making the strands easier to pull apart. In the laboratory, the strength of this interaction can be measured by finding the melting temperature Tm necessary to break half of the hydrogen bonds. When all the base pairs in a DNA double helix melt, the strands separate and exist in solution as two entirely independent molecules. These single-stranded DNA molecules have no single common shape, but some conformations are more stable than others. In humans, the total female diploid nuclear genome per cell extends for 6.37 GIGA base pairs (GBP), is 208.23 centimeters long and weighs 6.51 picograms (pg). Male values are 6.27 GBP, 205.00 centimeters, 6.41 pg. Each DNA polymer can contain hundreds of millions of nucleotides, such as in chromosome 1.
Chromosome 1 is the largest human chromosome with approximately 220 million base pairs, and would be 85 millimeters long if straightened. In eukaryotes, in addition to nuclear DNA, there is also mitochondrial DNA, mtDNA, which encodes certain proteins used by the mitochondria. The mtDNA is usually relatively small in comparison to the nuclear DNA. For example, the human mitochondrial DNA forms closed circular molecules, each of which contains DNA base pairs, with each such molecule normally containing a full set of the mitochondrial genes. Each human mitochondrion contains, on average, approximately 5 such mtDNA molecules. Each human cell contains approximately 100 mitochondria, giving a total number of mtDNA molecules per human cell of approximately 500. However, the amount of mitochondria per cell also varies by cell type, and an egg cell can contain 100,000 mitochondria, corresponding to up to 1,500,000 copies of the mitochondrial genome, constituting up to 90% of the DNA of the cell.
That is all for today, folks. One last thing. Before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Brooke Solomon, MSc in Genetics. We will see you in the next video. Goodbye.